Tre cose. La passo.
I don't know even how to play badminton, really. I don't know. And Saida, the favorite sport in Azerbaijan? While we are waiting for the people coming inside, of course. Saida?
Yeah, I'm trying to unmute myself. You know, we actually love a lot of sports, but I think racing kind of favorite one now, because we have it for many years, I think for 4 years or 5 years now. So we like watch racing. We also like football, but try to be as good as Italian. Not very well, but yeah, so a lot of things. Gymnastic is very famous in Azerbaijan too. Yeah.
Okay, thank you, Saida.
Okay.
We wait at least till 30 participants. A few minutes more.
Can I take the opportunity to ask Alexandru, how do I pronounce your name properly?
It's Alexandru, but you can always pronounce it Alex. Everybody just calls me Alex.
Alex, and your
Yeah.
surname?
Yeah, it's Vlaiculescu.
Vlaiculescu.
Yes, thank you. I know it is a bit of a...
It's fine, you just need to know it once.
Joyty, by the way, when it's your time, if you want to say something about CRC care activity and what is it, we did also for FAO and European Commission, other institution.
Just in our introduction, I suppose.
I have a question in regards to the session, actually. So if we want to ask the question from the participants, right, should we just write in the chat or ask them? And will you stop us and ask for the questions, and how many questions we would allow to ask?
We will see how it develops, the discussion, but at least we will have one question for each of you.
And we're also allowed to ask other participants, right?
Say that again, please.
And we're also allowed to ask the other participants.
Certainly, certainly. By chat, yeah, by chat we give the whole audience the opportunity to ask questions.
Okay. Should we raise the hands or like make a eye contact? How we can just stop and ask the question? Maybe we can just like, you know, so not unmute ourselves, but maybe we can just in the chat the question. How does it sound?
Yeah, indeed, everyone can place the questions on the chat. So then during the question session, we will distribute the questions according to who is talking. So yeah, if you have any question for the others, you can place them in the chat, and then we organize the session.
And Saida, if I may, if there is a quick question, yes or no, or something like that in the chat to you or to any other speakers, feel free to answer directly into the chat. If there is a complex thing, maybe better to wait to the question and answer session.
Great, thank you. That's clear now.
Michael, what about, we have 33 participants. What about when we leave, when we start 9:10, so 1 minute more? Or do you think we wait for 40?
You are the chairs.
Okay.
You will decide, it's your home. I'm just an attendant as any other.
Okay, well, I don't know about that. Okay, we start in 1 minute. because then we're 10 minutes after the official start. I think that's reasonable. So everybody concentrate. We start in 30 seconds. "35 participants. Okay, ladies and gentlemen, good morning, good afternoon, good evening, wherever you are in the world. A very warm welcome for session number 10 at another day at Remtech Europe 2020. This session is about the relation between organisms and contaminants in soil. And this relation is very complex and interesting. The first thing that comes in mind, at least in my mind, is that all these chemicals in soil, that they frustrate the organisms in soil massively. We tease the organisms with the pesticides and the industrial compounds. We hamper the functions, we even kill the organisms. The response of the organisms from this perspective is remarkable. They don't seem to blame us that we frustrate them. No, they help us. The worms, the nematodes, springtails, actinomycetes, bacteria, plants and trees, they all help us getting rid of the garbage in our soils, including these contaminants. And this is exactly where this session is about. It's about bioremediation techniques, and it is called a challenge for slightly contaminated soil and mine tailings. Before I shortly introduce myself, and also my two co-chairs will introduce themselves, I will ask you again, like we did yesterday, if you have questions in between, please put them on the chat. You know that we won't have questions after each presentation, so put them on the chat. And in the end, at the panel discussion, we try to come back at your questions. Okay, let me shortly introduce myself. I'm Frank Zwartjes from the National Institute for Public Health and the Environment in the Netherlands. And I deal with soil pollution as a project manager and also as a scientist, and I do all kinds of things that has to do with soil pollution. Natalia, would you be so kind to introduce yourself too?
Good morning, everyone. Thank you, Frank. I'm Natalia Rodriguez Eugenio from FAO. I'm working at the Global Soil Partnership. I'm dedicated to issues related to soil pollution. We are currently developing a global assessment on soil pollution, but also on other tasks related to sustainable soil management. Welcome to everybody to this session 10.
Jyoti, the word is for you.
Unmute myself first. So I am Jyotish Najid. I am a program leader in CRC Care. For the past 7 years, I have been developing Australia's national framework and guidance on site contamination. This has been possible through the partnership of government, industry, and universities which CRC Care represents. So CRC Care actually stands for Cooperative Research Centre for Contamination Assessment and Remediation of the Environment. It is a center of excellence for conducting collaborative research. My role has usually been in policy, and one of our key outcomes has been the development of a national remediation framework. It's one of the first that's been around, so we were quite excited about that. I'm also excited to be here today, co-chairing this key session on bioremediation, and I believe we'll cover phyto-remediation as well. Both are very sustainable methods for conducting remediation. We will hear from today's speakers more about their projects, and we are looking forward to it. Thank you. Over to you, Frank.
Thank you very much, Jyoti. We have 6 interesting presentations ahead, and Natalia, may I ask you to announce the first presenter for this session?
Yes, indeed. The session then starts with a very interesting presentation from all over the other side of the world. We are traveling to Malaysia, and we will hear Jejani Vijanathan from Forest Research Institute of Malaysia. Her presentation is titled Acacia Magnum as a nature-based solution for bioremediation of extinct tailings in the tropics. So, Jejani, the floor is yours.
Thank you very much, Natalia. Let me share my presentations. Hold on. I'm having trouble. Just give me a minute.
Sure.
Hold on, my slides.
No, we cannot see them yet.
Can you see? No.
No. Share the screen and...
Yeah, I had it here. I was able to share it just now. I'm able to see it now.
It did work 5 minutes ago.
Yeah, it did. It did work. I'm trying to see how I can share. I can't see my files. Hold on.
The green button is not responding.
Yes. It is not in the dashboard. Sorry.
Okay, now.
Can you see anything?
Yeah, now we see your desktop.
You see my desktop. Okay, yeah. Hold on. It's here. I'm sorry. Okay. Can you see it now?
Yeah, I think it's going to work.
It's loading.
Okay.
Just remember to put the presentation mode.
Okay.
Excellent.
Okay, perfect.
Is it okay?
Yes,
Very
perfect.
sorry for the delay.
No.
Okay. All right. Good morning over there and good afternoon for those in Malaysia. I'm Jeni Vijanathan. I would like to share my presentation titled Acacia Magnum as a nature-based solution for bioremediation of extinct tailings. I'm from the Forest Research Institute of Malaysia, and this paper was co-authored with my previous director, Dr. Ahmad Sohaidi. So firstly, I would like to share some of the issues in the mining areas in Malaysia, mainly on the industries, the economic importance, as well as the environmental degradations happening in extinct tailings. Then we will move on into the definitions of nature-based solutions and how this strategy can be used for ex-mining areas. We will move on to the area of interest, area in the north side of Peninsular Malaysia, as well as the methods used. Before I briefly explain the results, discussion, as well as the summary of our work. So basically, what do we have in Malaysia is that most of the areas that used to be mining has actually dwindled. I'll show that in the next slide. We have same issues as how that you all are facing, whereby mining areas go through land degradation, erosion, sinkhole formation, as well as loss of forest and biodiversity, not forgetting the potentially toxic elements which are also available in ex-mining areas. We all know that these PTEs are essential, but when it's in high quantities, it's quite detrimental for humans as well as plants. Soil pH is a major factor which actually affects the availability of PTE. When it's very acidic, we can have very high levels of PTE, besides reducing conditions, as well as others such as macronutrient levels, water, and temperature. So, excuse me, the curve that you see here actually shows you that in Malaysia, the mining area was close to about 300,000 hectares, excuse me, in 1970, and it quickly dwindled, probably because of the some of the issues related to the crash of the tin mining industry in 1985. You can see it was merely about 26,000 in 2017, and this data was between a gap of 20 years. So only in 2007, the Environmental Act came up with some regulation regarding rehabilitation of ex-mining lands with forestry species as a pollution control method. Of course, it is cheaper, but it takes a lot of time to establish forests on a bare land or a degraded land. So nature-based solution, while some of the main goals here is to protect, sustainably manage, and restore natural or modified ecosystem, this has been coined by the IUCN in 2016. Has been used well in climate change, as well as in food security, and for protection of conservation areas. There are two papers which you can look up by the same authors here, Kistra. The first paper on soil-related sustainable development actually talks about the different types of land degradation, may it be physical, chemical, or even biological, and how sustainable development goals are being used to remediate. The second one speaks about more on landscape solutions as well as soil solutions, and how we can fit in nature-based solutions into remediating areas which are heavily degraded. So we chose a species which is quite common to us, Acacia mendium, because there's been various studies showing that acacias are actually able to absorb PTEs in their system, as well as they seem to grow robustly. They have dense foliage with high litter inputs, and also it's a non-food crop. So the issue of food crops being planted in ex-mining area would not be an issue. So if you look at the graph here are some of the processes related to phyto-remediation. Work by previous workers have shown that Acacia mendium is able to stabilize through phytostabilization, as well as do phyto-extraction of PTs in its system, in the plant system. The table here that I am sharing with you is actually some of our previous work in 2017. What we found was that the amount of iron, aluminium, as well as zinc was very high in the plant parts of Acacia mendium when we firstly planted. And then the other PTs were either low or in negligible values. So this is the area that we were looking at. There was an ex-mining area. It is still ongoing. It's known as Perak. I think Chuck can relate to it because Perak means tin. The whole state used to be one of the major states producing tin ores in Malaysia in the early 1970s. And the name also carries the name Perak, means tin in Malay. So we went into this area. We were approached by the miners in early 2010 in order to rehabilitate back in this area, and we only started planting Acacia mendium in late December 2011. So, so this was the area which was mined. Okay, some of the pictures that you can see here was that these were the Acacia mendium seedlings, about 3 months old, were planted in the late December 2011, we had sub-trials within the plot whereby we had those applying compost. Another one was just with the seedlings alone. And after 5 years, in 2016, we went into the area bank to do soil sampling, mainly to see how much of heavy metal was in the soil, as well as how much it has been absorbed by the trees. So we did some destructive sampling. We collected the roots, the wood discs, the foliage, and then we ran some tests in the lab using ACKNOWLEDGEMENTS method to determine the availability of PTEs. So this table actually shows you the amount of bioconcentration factor as well as translocation factor that we got from the 4-year-old acacia mendium trees. The BCF is actually the concentration ratio of roots to soil, and the TF is the ratio of heavy metal to the aerial parts to roots. We found that the TF for the iron, aluminium, as well as zinc was more than 1, fitting to be a metal accumulator compared to the others which we tested. For bioconcentration factor, there were only some high values for sulfur detected. So some work have shown that usually phytoaccumulators are noted to have high TF and BCF, but our study was similar to what was reported by Nick et al. 2012, whereby this particular species had high TF but had low BCF. And we believe that this was due to the acidity of soils. It was quite, it had very low pH, about 4 or 3, and probably because of that, some of the elements that we were hoping to be absorbed by the tree may have bind with other elements in the soil. So particularly, although this project is still ongoing, some of the key points that I would like to share is that planting acacias actually allowed high litter inputs to rejuvenate back the soil, allowing to enrich the organic matter. Of course, we know the acacia mendium is from the family Leguminiaceae, and it has nitrogen fixing bacteria, and the root proliferation may also host mycorrhizas. And basically in Malaysia, one of the major economic importance of this species is mainly for pulp and paper rather than for furniture or other timber industries. But slowly, some of them are also using this species for furniture making. So we like to summarize saying that it is a potential species for NBS in afforestation and as well as to mitigate soil pollution. This slide actually just shows you some of the key points which have been outlined in the voluntary guideline on sustainable soil management, and how some of the work that we are doing actually also echoes the same thing here. We hope that the establishment of new forests would actually allow some income generation, biodiversity enhancement, and especially for the environmental portion of this area. So some updates that I would like to share with you is that besides acacia mendium, which was actually planted as nurseries, we have also introduced other timber species, and we are going to look at how they are going to grow under this host species. And we'll be doing further soil as well as plant sampling scheduled for November 2020, in about 2 months' time, to see if there's any changes in acacia mendium as well as the species that we have introduced there. We also think that the work that we are doing is able to address some of the sustainable development goals as numbered here: 6, 12, 13, 15, and 17. So this is my last slide. The picture that you see here is actually the 10-year-old acacias with the Shorea roxburghi species. And you can see inlet how it was before we did some work and how it is now. So we like to thank Prim as well as the ex-mining company, the tin mining company, as well as Remtech Expo for inviting me to speak today. Thank you. Razi.
Thank you so much, Dejani. Very, very interesting and amazing to see the result. I give now the floor to my colleague Jyoti for introduce the next speaker. Thank you.
Thank you, Natalie and Jani. We're still in Malaysia, and it is my pleasure to introduce Chuck Chuan, whose topic is enhanced cadmium-led phyto-remediation in vetiver grass using different soil amendments. Over to you, Chuck.
Hi, very good day, everyone. Are you seeing my screen? Can someone just...
Yes.
And can you hear me? Yep. All right. That's great. All right. All right. Okay. I think without further ado, a very good day once again. And let me just start by introducing myself. I'm Chuck Chuan. Okay. I'm from the China ASEAN College of Marine Sciences from Xiamen University, Malaysia. And the topic today I will be talking about is the enhanced cadmium-led phyto-remediation in vetiver grass using different soil amendments. So similar to the first speaker, Jani, she actually shared almost similar research areas with me, unlike she use Acacia mengium, and I will use Vetiveria over here. So without further ado, let me just talk about my content of presentations. I will start with a big background and also the significance of this research. Okay, what are the research objectives? What are the methodology used in this research? And I'll go to the most critical part of the research, which is the findings, and followed by the conclusions and some part of the acknowledgement, and also a list of publications and some real sites plantations of the plants, and also some references. And if we have time, we'll go for a Q&A at the end. All right, so in terms of the background and the significance of the research, as what the first speaker have said, in Malaysia itself, we have all sorts of soil pollution. So we could not run away from that. And most of these soil pollution are caused due to our human factors. And it is an anthropogenic factor, such as from the industrial, as well as from our domestic household activities. And because of that, we have various remediation technologies, as most of you are aware of. We have the physical, we also have the chemical assisted remediation techniques, but most of them are either non-cost effective and non-economical, as well as some of them are environmentally intrusive. That is why we were very interested, both of us, Joanny and myself, and are interested in phyto-remediations, which is a part of bioremediations. Because with the use of phyto-remediation, it is a type of green plants, a choice of a green plants used to clean up, or in other words, to metabolize or degrade soil heavy metals with the minimum cost, and also at the same time, it is environmentally friendly. So let's just go back to the basic, what is phyto-remediation? So phyto-remediation is a sub-component from bioremediation. It uses the biological process to either degrade, break down, transform, or essentially remove any part of contaminants that we do not want in the soil or water. So phytoremediation, in other words, is a form of bioremediation, which it uses plants. And as for my case, I'm using vetiver. So if you look into the Greek, the breakdown of the words, phyto is derived from the Greek words mean plants, and remediation is basically from the word remedium. It talks about restoration or cleanup. All right, and before we look into the most areas of phytoremediation, we also look into subset of some phytoremediation. It can be subdivided into more than 5, but over here I just try to list the 5 common types of categorization of phytoremediations. It is also cover phytovolatilization, phytoextractions, phytodegradations, phytostabilization, as well as rhizofiltrations. Every part of the plants have a different functions which helps to clean up the contaminants in the soil. And for my research, I cover mostly on heavy metals, which is cadmium and lead to be specific. So the main extraction of having phytoremediations are a lot, and just to name a few, it includes, as what I mentioned earlier, it is cost effective and also economically efficient. It is also locally available if we choose the right plant species. for my case is Vetiveria over here, or Vetiver in short name. And it is feasible to be practiced. Okay, at the same time, planting trees of any type of plants is simple approach and it can be easy to manage. That is some of the main attractions of phytoremediation. So back to basic, what is vetiver grass, or the scientific name known as Vetiveria zizanodus, is a most promising plants as what I have conducted research on other plants as well. So it is a fast-growing plants. It have high resilience to many environmental stresses, and it have high tolerate to extreme metal condition, especially heavy metals. So most of the past studies actually focus on single metal accumulations. So what I try to be different in these particular studies is I try to look into the combination of both cadmium and lead, because both of these are highly contaminated and yet it is hazardous to human health as well. So that's why I'm looking into these two elements. So both of these cadmium and leads are also easily exposed through direct inhalations and indigestion of soil, and ultimately it also affects human health, which can be fatal. So that is why in order to increase the metal accumulations, I also look into how can I incorporate to enhance the availability of the accumulation in the plants, in this case vetiver. So I look into various types. I look into using the chemical assisted properties such as EDTA, which is the salt, and I also use the sulfur, which is commonly used as a micronutrient, and also an fertilizer, which is a common use as a fertilizer for most farmers. So the objective of these studies are mainly two. I would like to look into the trends and effects of the heavy metals that I'm testing, and at the same time, just look into what are the influence and the capability of different types and levels of those three soil amendments over here: EDTA, the sulfur, as well as the ammonium nitrates, or in other words, the N fertilizer to be short, in terms of the plant species growing under these mixed cadmium and lead contaminated soil conditions. So in terms of the soil methodology, so I have the preliminary soil assessments, and I do the artificial spiking, and I do spike the soil up to the contaminated level based on the Malaysian Department of Environment standards, as well as following to the Canadian Council of the permissible level. So my cadmium is spiked to 50 milligram per kilogram, and my lead is up to 100 milligram per kilogram. So, and I also use the flame atomic absorption spectrometer to detect my concentration, as well as I use the standard reference material from Germany to ensure the precision of the technique. So in short, you're seeing a list of treatments that I'm having in this study. Basically, I have 4 different levels of EDTA with one EDTA as well, which is the 1 millimole per kilogram of soil, 5 millimole, and subsequently to 10 and 25. And for elemental sulfur, I have 5 treatments, 5 different treatments level, and subsequently for nitrogen fertilizer, I have 6 different treatments. And this is the descriptions of all the treatments I have, and all are with 3 replications. Okay, so it is tested in a pot assays conducted under a completely randomized design in the plant house, and I do have certain parameters very similar to what the first speaker have used as well, using TF and BCF. I also have other like method efficacy to detect the outcome. So what are the outcomes? Before that, there are some pictures to show you the plant experiment process. This is the uprooted version of the plants. Okay, so this comes to the research findings. So basically in the research finding overall, in terms of the plant growth, especially in terms of plant heights, I will only highlight the significant value or the significant results that I found. So in terms of the 40 millimole of sulfur that we use, it shows significant growth. Okay, it's a kind of a surprising that only this particular element show a very significant growth. And on the other hand, no doubt, as it is a fertilizer, nitrogen fertilizer, the highest use of the nitrogen, it has the highest plant growth. Okay, there's no other significant in basal diameter. And now we look into the accumulations in terms of cadmium. In terms of cadmium, it is surprising to see that in the shoot itself, when we use more EDTA, it actually accumulates more in the shoots, okay, of the vetiver grass. But on the other hand, It is also similarly high when we have the 10 millimole and also 20 millimole of sulfur. And subsequently, the N fertilizer are also high when we use more N fertilizer. So this is the outcome that we receive. But in overall total, the more EDTA we use, the better the overall results. Because as you can see, those in reds, the roots actually have a decreased accumulation as it increases. So this is the trend. And on the other hand, when we look at the lead, this is the lead, table 4 shows the lead accumulation level. Both elemental S and N fertilizer show significant reduction. The more the use of elemental S and also nitrogen fertilizer, the result actually is not positive. So it means it actually shows an opposite trend. While on the other hand, EDTA for lead is still increasing in the shoots, means phyto extraction is working over here. So this is the results that we obtain from the research studies that we have conducted. So, and if you look into the comparison of TF as well as the BAC, as well as the most importantly, the efficacy level or the percentage of efficacy. So in summary, we can actually find that the 25 millimole of EDTA accumulated the best or the highest among everything, where it comes to a total of 63%. Okay, and that shows significant to the control and to other treatments as well. On the other hand, when we look at the among all types of elemental S, the 20 millimole of sulfur actually accumulated the highest as compared to the others. And of course, subsequently, in terms of still cadmium accumulation, the highest 300 millimole of nitrogens accumulate the highest. So the trend is a bit different when we look into the lead now, as a while ago was cadmium. So 25 millimole of EDTA still accumulate the highest, 10 mole for sulfur accumulate the highest, and on this case, nitrogen fertilizer was not the 300 millimole, but it's the 10. So what can we conclude over here is based on if we conduct the regression studies and also look into the trend, basically in overall, the general experiment finding shows that cadmium was remarkably recorded high in the accumulation of vetiver under a mixed contaminations. So in overall, the use of EDTA is very promising, especially with the 25 millimole, followed by the subsequently from using elemental S, sulfur, as well as the N fertilizer. So in short, we can see the records that N fertilizer and also EDTA have the significant results in the following orders. The best that we can use is EDTA, which has also proven in literature works the best, but under a mixed condition, it is still the best, which is work the best, followed by N fertilizer, which is less effective, and also sulfur, which is the least effective over here. This is the overall general findings that I found. So in conclusions, vetiver grass is relatively a good tolerance because under such high condition of contaminants, it's still able to survive and able to accumulate high level of contaminants, especially lead and cadmium. So in short, the effectiveness are as follow. EDTA work the best, followed by N fertilizer, and subsequently the least is elemental S, sulfur, under the mixed cadmium lead soil conditions. So with that, I would like to just acknowledge the funding that I researched for this project is from the University of Malaya. And also I would like to thank Ramtech for the opportunity. And also for this particular project, the paper has been actually published back in 2006. And I would like to re-acknowledge that there were also subsequent project running as well. And there was another paper published, it's also 2006 in other journals. And aside of just doing a plant house experiment, we also conducted the projects on site. We carried out for parts like 6 months plus, and subsequently the site was disrupted. So we could not able to analyze the results. But in overall, we still are working towards other similar research. And with that, I would like to end the topic of enhanced cadmium lead phyto-remediation in vetiver grass using different soil amendments. I think that's all from my side. Thank you very much.
Thank you very much, Chuck. That was a very interesting presentation. Mixed contamination is a very practical way to look at things because in a lot of situations there are mixed contamination. So that's very, very interesting. Unfortunate that you couldn't complete your research on site. It'll be interesting to see the results of those at some point. Yes, I'll pass on to Natalie to introduce the next speaker, please.
Thank you, Jyoti. Now we travel to Azerbaijan. We have with us today Saida Alijeva, who will present from the Azerbaijan National Academy of Science, who will present the work development of biotechnologies for cleaning of irrigation canals from oil pollution and heavy metals. So indeed, it also seems very interesting and talking about mixed pollution. Dear Saida, the floor is yours.
Yeah, hi, thank you very much for the introduction. Yeah, so I'm gonna present today the information. Oops, let me go back actually. So let me see if I can go back. Okay, yeah, so I'm gonna talk today about the new biotechnology that's used for cleaning of different type of pollution, the organic pollution and also the heavy metals. And I just try to go back and show the first screen, but I don't know if I can do that. Okay, yeah, here we go. So the my co-author, Professor Nariman Ismailov, worked in this issue for many years, and we are trying to address one of the main important problems related to the irrigation channels. And let me start with the main problem that the highest water consumption related to the agricultural irrigation in the most countries of the former Soviet Union. It's about like 23 million of hectares, and it's definitely varied in between the countries. Depends on their condition, surely. And the most important step that in Caucasus area, the range of the water used for the irrigation is varied in between 40% up to 80%. In Azerbaijan in particular, we have about 1,350,000 hectares that are irrigated areas, but that's not the main problem. And even the using 60 to 70% of all water resource for the irrigation is still not the major problem. So what is the major problem? We have two major rivers in Azerbaijan. One is Kura and the second one is Aras. And this Kura-Aras river system is internationally significant, but at the same time, it is under the threat. The threat of the water scarcity and the threat of the pollution. We cannot, in our research, solve the water scarcity problem, but we try to address the pollution problem. Let's move forward. Here on the map, you can see actually the river system. I did not include the country's name, but I want to mention that Kura River, which is the largest one for us, located in the territory of 4 countries, 4 border countries. And the 40% of the population and the most big industrial potential located just close the territory of the Kura River. So as a result, what's happening? We have all this pollution inside of this Kura River. So what next? The Kura River flows through the, take all this pollution internally and through the border countries, and all this pollution flew to the Caspian Sea, which is really rare and unique reservoir. So we need to save the ecosystem of Caspian Sea. But what kind of pollution do we have inside of that? And I can mention all of them, but let's just, I will just name them as organic pollutants, all of them. And the source for those organic pollutants are really different. So here on the table, you can see actually the territories of 3 main countries located on the border of the Caspian Sea. And in the perspective of Azerbaijan, we have this Kura River, which, through this Kura River, we have oil products in amount of 3,000 tons per year that flows into the Caspian Sea. So taking into account the importance of cleaning these, rivers of the pollution, we start developing our biotechnology. And the problem with the contamination is also going, not only related to the Caspian Sea. Again, we have another problem. We have this pollution into the soil. And as a result, we have these phytotoxin products. So and the phytotoxicity of the soil increased. For example, if there are river floods, then the pesticides and herbicides actually wash out from the agricultural lands and also goes in, runoff to the Caspian Sea. Another problem is that we have this 20 years of more than 20 years pollution with the petroleum hydrocarbons into the Caspian Sea. Even we have a lot of measures to prevent that. However, it's still ongoing. So the main pollution source for let's go back to the Kura River, because we are trying to prevent the pollution before it's entered into the Caspian Sea. And the main pollution source are household, industrial, and agriculture wastewater. So that's the three priority that we try to address in our research. And so what we have, the main goal of our study is developing the novel biotechnology, which we bind to the application method that we can apply in the main canals, irrigation canals, such as Upper Karabakh and Upper Shirvan canals. And it's really crucial, for enhancement of the agricultural product quality and also for preventing the soil from the contamination with these organic pollutants and also heavy metals. So what we did, I'm not going to go too deep in the microbiological stuff, but I will just explain the most important part. So we developed the biopreparation using the microbial consortium from our institute collection. We have the microbes that could pretty good eat the organic pollutants, such as pesticides, such as oil products or oil hydrocarbons, all the oil hydrocarbons. So we make the great consortium that work in regards to the degradation of this organic stuff. And at the same time, we had the molecules of the bacteria that could absorb the heavy metals on their cells. So we have the great consortium. Then we have this biopreparation, which we prepared as a biofilm. And then we also use the sawdust and also the active sludge. So we can have the active sludge for any, from the cleaning factories, from the municipal waste. And so we collect this active sludge, sawdust, and biofilm all together. And now we have the biopreparation that could use perfectly. We tried in the laboratory, it works. What we did next, we developed the laboratory model of the irrigation channel. So we tried to mimic all the shape and the format of these irrigation channels in the laboratory. And what helps us, we actually cover the internal walls and the bottom of this irrigation laboratory canals, laboratory model with the microfiber fabrics wrapped. So we just wrap it. And what happened next, that our biopreparation greatly absorbed by this microfiber. And the next step that we have is our biofilm that pretty great work inside of this microfiber. And here on the picture, you actually can see these, the biofilm formation process that we have and sawdust that we use. And then as a result, we have these biobonds. And these biobonds absorbed by these microfibers, as I mentioned. And then the next stage, let's see the results. It works. About 80–10% of the organic pollutants just disappear after we use it for the certain time. And so what happened that our biopreparation work as a biological filter. So it filter it actually collect the pollution and then decompose it. And what we like that this microfiber fabric absorb the pollutants along with the biopreparation. So they work together inside of this microfabric, and then the interesting thing that they didn't wash out. Why? We ask ourselves why? Because the periphytons collected there too, and they work all together. And so they kind of create a natural self-clearing condition for these irrigation canals. what we also recommend for using this our biopreparation, our biological filters, we call them now, that they should be located along the irrigation canals, and they should be in their layers. In with in between them, they should be like 10-30 meters, and the different levels. Why? Because we don't want to have flow rate reduction, and we don't want to impact the ecosystem by this water flow reduction. So nothing will change in this actual reduction in regards to the water flow. That's very important for us because we don't want to touch the ecosystem itself. And another thing that and if you see in this diagram, we can reduction of the heavy metals in this water after using our biopreparation. So we definitely recommend practical application of this technology. And we're going to talk with the relevant organization. I hope they would be interested in that. Definitely, this is the great way towards the green economy. First of all, we will have clean products, agricultural products, right? And at the same time, clean water that could be applied to grow this agricultural products. Moreover, this method actually requires the special conditions, so the special temperature, water composition, wind. We need to consider all those components, all those conditions when we apply our biopreparation. So what we plan to do, and we start working on that, we're working on the computer program that would help us to estimate and predict the best position of the biofilters in the irrigation canals. That's really all. We are still working on this project. Thank you very much for your attention.
Thank you very much, Zaida. Indeed, very surprising and of course challenging, because controlling a living organism is not that easy, but it seems very promising. Thank you so much. I leave the floor to my colleague, Jyoti. Thank you.
Thanks, Natalie and Zaida. I am in mute. Yep. So it is my pleasure now to introduce Alexandra Vlaiculescu, and his topic is bioremediation of the herbicide DICAMBA, utilizing pure cultures and microbial consortia associated with biological control against the fungal pathogen Fusarium calmorum. Over to you, Alex.
Yes, hello. Can you see my screen?
Yes, you just need to. Yep, excellent.
There we go. Hello. My name is Alex, and the title of this presentation is going to be Bioremediation of the herbicide DICAMBA associated with biological control, a possible approach. And I'm going to start with, this is the outlook of my presentation that will be following, starting with a quick introduction of the concept that we utilize and moving on to the results and, of course, the conclusion. First of all, we're going to start off with what a pesticide actually is. And that is a chemical compound that is used to prevent, destroy, repel, or mitigate pests. Pests, which can include insects, rodents, fungi, and weeds. And on the right, we have a table that we can see benefits and risks associated with the use of pesticides. We have the main function of pesticides, which is crop protection, but they also offer food and material preservation and disease control. Unfortunately, they may present high toxicity to humans. They can have a very strong impact on the environment, and they're also prone to bioaccumulation. Now, the pesticide that we used in this study is called DICAMBA and is a benzoic acid herbicide that is used to control annual and perennial broadleaf weeds in grain crops and grasslands. It is highly soluble in water, which is actually an issue because it has a very easy time of actually leaching into groundwaters and streams. Around 2016, its use came under significant scrutiny due to the fact that it has a tendency to spread into neighboring fields, causing damage. And this is especially true for very sensitive crops, such as soybean, as you can see on the picture on the right. Concept that starts from the beginning of this presentation, and as you're well aware, bioremediation is a process that utilizes microorganisms or their products in order to treat polluted sites with the final goal of restoring them to their original condition. There are multiple types of bioremediation, of which we emphasize bioaugmentation, which is based on the inoculation of given environments with microorganisms that possess certain capabilities. In our case, what we're looking for is, of course, pesticide degraders. So could microbial degradation be the solution for this? We have a few examples on the table on the right of some common pesticide degraders and the pesticides that they were able to degrade in literature. And we can mention several pesticide degrading genera, of which Pseudomonas, Bacillus, and Flavobacterium. Since we discussed pests such as weeds, what about fungal pathogens? Well, we know now that it's estimated that about 25% of global cereal production may be contaminated with mycotoxins. And this raises the need for control of fungal pathogens. Of those, we can mention the genus Fusarium, with Fusarium culmorum as a representative that can cause Fusarium head blight and Fusarium seedling blight in wheat, barley, and oat. And we have examples of both diseases at the bottom in wheat. And Fusarium culmorum is also the fungal pathogen that we chose as a model in order to test the biological control function of our organisms. And moving on to our scientific question, we can now state that pesticides are compounds used to control different pests, but at the same time, they present several risks to sensitive crops, humans, and the environment. Well, companies are trying to develop products that would act as biopesticides and provide benefits to the crops and soil. For example, Christian Hansen has been working with species of the Bacillus genus, and they have been releasing products such as Quartzo and Presence, which are bionematicides, so they're used to control nematode population, as well as Intense, which is a biofertilizer that promotes the health of the plant and helps against stress. Well, we can clearly observe a trend in developing plant protection products from microorganisms. So what if we could find a way to degrade the existing soil accumulated pesticides, while at the same time provide with other pest control alternatives? And applying that specifically to our situation, could we find bacteria that degrade the pesticide DICAMBA, while at the same time they have biofungicide abilities against Fusarium culmorum? In order to be able to achieve that, the two functions are tested separately. So pesticide degradation is tested in liquid and solid medium, while crop protection function or biocontrol is tested in some wheat seedling setups in sand pots that are inoculated with the fungal pathogen. Organisms that prove abilities to exert this dual function ideally will be tested in a coupled experiment. As the title of the presentation suggests, this has been a two-part project performed in collaboration with University of Copenhagen. So the part that concerns pesticide degradation is on the left and has been performed in Aalborg University, while the part that concerns plant protection has been performed in University of Copenhagen. And moving on now to our experimental setup, the first experiment that we performed was the pesticide degradation assay, in which the degradation efficiency of about 60 previously isolated strains that originate from soil and sludge that were enriched on pesticides. It was assessed through a 2-week experiment. 50 milligrams per liter of DICAMBA was used as the sole carbon source, and the determined amount has been, the degraded amount has been determined through HPLC analysis. In parallel, we also performed an enrichment of the mixed cultures, full mixed cultures for that matter, from pesticide contaminated soil samples, and the 5 grams of soil were added into 20 mils of enhanced M9 medium, a medium that I will describe a bit later. And the cultures were passed every 7 days in order to wash out the soil and leave the pesticide as the carbon source. Moving on, we also performed a solid medium assay in which the cultures were plated on a variation of the M9 medium with DICAMBA. And this was used in order to expedite the screening and avoid the preactivation of the frozen cultures in a more nutritive medium. ATP kit measurements were taken using the Bactiter Glow kit from Promega in order to determine the metabolic activity of our cultures at different times. As for the experiments performed in KU, we used a wheat seedling assay in which wheat seeds coated with the fungal pathogen Fusarium culmorum and possible biological control agents, or bacteria, are placed in sand pots and incubated for 2 weeks in a growth chamber. After those 2 weeks, the severity of Fusarium seedling blight symptoms is assessed, and the second type of assay was also performed, a seedling assay that contains no biological control agents, and at the same time, it contains no fungal pathogen, only different concentrations of DICAMBA. And this has been done in order to see what different concentrations of DICAMBA could have as an effect on the seedlings, so we can then move on and combine this and add it into the big Fusarium seedling assay. Now, for the pesticide degradation assay, 6 batches of organisms were tested, usually represented by 10 organisms at a time in order to be able to keep the conditions as homogeneous as possible. We observed initial inaccuracies, mainly due to calculation of evaporation during the 2 weeks of the experiment, that were solved until the end. And the size of the error bars is due to the propagation of the error with subtractions greatly increasing it. As a result of this assay, 6 possible degraders were selected, and the ones that have been shown in the graph. As for the nomenclature of the bacteria, they all start with the soil sample that they originate from, so HS from Hungarian soil, FS from FMC soil, and SLU from sludge. They go on with the pesticide that they have been enriched on, so DICAMBA, dimethoate, or malathion, and the randomly assigned number. And this we have an exception for the organism Nolga, which is a Klebsiella strain. As this seemed to be a bottleneck in bacterial growth, medium optimization was the next step that we performed. And since the medium used in this study was M9 minimal medium with 50 milligrams per liter of DICAMBA as carbon source, this might not fit the necessary requirements for most organisms. So an improved medium has been devised, one that contains amino acids, vitamins, and trace minerals, the ones shown in the figure. And this will be further referred to as the enhanced M9 medium. And it is the one that has been further used in the next experiment as well. For the enrichments now, 4 different mixed microbial consortia were isolated from 4 soil samples, and the cultures carry the name of the samples. Again, the cultures were passed every 7 days in order to wash out the soil, and ATP kit measurements were used to determine their state at given times. The graphs at the bottom represent the number of light units, which is the unit of measurement for the ATP reaction of 2 of the passages, along with the dates of the measurements. And you can see that in these passages, the organisms that seem to thrive are HS and FMC West, which have the highest value from the beginning of passage 2 and all the way until the end of passage 3. Now, moving on to passage 4, the last one that we performed, the cultures shift, and the one with the highest value now is FMC-MP, as you can see, followed by HS. And this could be due to a multitude of factors, but mostly possibly to the washout of the soil and the absence of a secondary carbon source, suggesting that the cultures that thrive now are actually the ones that are able to survive on DICAMBA alone. For the plate assay, enhanced M9 medium with 50 milligrams per liter of DICAMBA was used, and the organisms were plated and their growth was assessed after 7 days of incubation. The situation unfortunately allowed for a small number of organisms to be tested, but 4 of them managed to grow on the medium. And in the pictures you can see Pseudomonas putida on the left and Micrococcus luteus on the right. But unfortunately, due to the closure of the university related to COVID-19, the method to quantify DICAMBA from the solid medium was devised, but not verified. A valid situation at that point was that the organisms might be feeding on something else rather than the DICAMBA. So for example, they could be feeding off the agar. So a medium that contains all the components of the enhanced M9 agar, but without the addition of DICAMBA has been devised. And the organisms were plated again on this medium, and they were treated exactly the same under the same conditions. And as you can see, they did not manage to grow, as you can see at the end of the picture. So that means they probably were feeding on the pesticide. Now, moving on to the seedling assay, the severity of the Fusarium seedling blight symptoms is determined through a unit called the disease index, which is calculated from the severity of the symptoms of the seedlings by scoring them from 1 to 4. The pictures at the bottom, you can see the seedlings after the 2 weeks of their growth. And in this experiment, Pseudomonas putida KT2440 was used as biological control agent at an optical density of 1 or 0.1. Unfortunately, the decrease in disease symptoms in these conditions has not been statistically significant. When plotting the germination rate, we could see that Pseudomonas putida did not affect the germination rate of the wheat seedlings in any way. While moving on to the second experiment, this time 3 different organisms were used as biological control agents, Bacillus subtilis, Micrococcus luteus, and a different strain of Pseudomonas putida this time. And the seeds were coated with a suspension of the bacterium at an optical density of just 1. As for quantifiable data, unfortunately, in this case as well, even though it seems that Micrococcus luteus and Pseudomonas putida at the end of the graph were both, it seems like they were both able to actually decrease the disease symptoms. Unfortunately, the results were not statistically significant, with p-values of 0.06 and 0.12 respectively. When plotting the germination rate in this case as well, the Fusarium-only condition exhibited a statistically significant decrease in the germination rate compared to the control sample, but that was to be expected. And aside from that, the rest of the conditions show no significant decrease in germination rate with the application of any of our biological control agents. Now, for the experiment that concerns DICAMBA application alone, 4 different concentrations were used ranging from 6.25 milligrams per liter up until 50 milligrams per liter, with 12.5 simulating field conditions. And as you can see, application of DICAMBA after 2 weeks had severe effects on most conditions. The only one that only suffered mild effects seems to be the 6.25 milligrams per liter condition. Now, moving on to quantifiable data, we plotted the shoot length of the wheat seedlings, and we could see that with the increase in dicamba concentration, there is a significant decrease in shoot length aside from the 6.25 milligrams per liter. As such, this seems to be the only viable dicamba concentration, since it would not greatly interfere with the assessment of the fusarium seedling blight symptom that we usually do. When germination rate was plotted, it came out as a surprise that even though the effect of dicamba on shoot and root length was severe, it did not interfere in any way with the germination rate. Due to the closure of the laboratories caused by COVID-19, many questions remain open. As such, the pesticide degradation assay can be improved in many ways, mostly by increasing the working volume and the duration of up to 2 months in order to allow for more degradation to actually occur. The MMCs, for them, since no further experimentation was actually performed, they should be tested using the pesticide degradation assay and the seedling assay in order to be properly vetted. The solid medium assay was also established, but at this moment, the quantification method from solids was not tested since the recovery rate of dicamba could not be determined in time. A coupled experiment is to be performed. That would be a combination of the 2 plant experiments shown, and it will contain all the elements of the fusarium seedling assay and the viable concentration of dicamba that we determined. Many more organisms remain to be tested using the seedling assay, preferably, ideally, all the pesticide degraders that we found. In conclusion, our main goal was to find organisms that can degrade dicamba and at the same time provide biological control against Fusarium culmorum, that which we call a dual function. For the dicamba degradation part, Bacillus subtilis, Pseudomonas putida, and Mycococcus luteus and FS mal 132, these were all able to grow on the solid medium, but so they were able to degrade dicamba, but the degradation could not be quantified at this point. As for the biological control, Mycococcus luteus and Pseudomonas putida seem to be able to decrease the symptoms, but not significantly. As such, we were unable to determine the dual function of our organisms just yet, due to the closure, which means that the screening will need to continue. And in the end, I would like to thank Professor Cristiano Varone for his immense support and Professor Birgit Janssen and KU for the collaboration on the plant experiments. And thank you for having me.
Thank you very much, Alex. You've covered a lot over there. I hope there will be some questions for you as well, so you can tell us a little bit more. I'll pass on to Natalia to move on to the next speaker, please.
Thank you, Jyoti. Well, we are now having someone from Libya, but unfortunately, because the connection was not good, we will see her presentation on a video. So Marco will put the video recording, and her name is Al-Halba Al-Hakima from the Libyan Petroleum Institute.
Yes. Al-Talba is sorry, she was not able to join us, and with showing this video, we also hope for a peaceful future for Libya. It's a priority also for all European countries. So let me show the video. Natalia, tell me in the first 15 seconds if it's something that doesn't work. Just a moment.
Sure. So the name of the presentation, while Marco put the video, is Potential of Ozonation and Phyto-remediation to Reduce Petroleum Hydrocarbon Levels Remaining After Pilot-Scale Soil Bioremediation.
Greetings from Tripoli, Libya. This slide shows a brief biography of myself. My name is Hakima. Potential for ozonation and phyto-remediation to reduce petroleum hydrocarbon levels remaining after pilot-scale soil bioremediation. Petroleum contamination is a major problem worldwide, dangerous to the human health and environment. Severe environmental contamination in Libyan oil and gas industry. TBH are one of the most common groups of resistant organic contaminants and are known to be toxic to many organisms. TBH affect soil quality due to their toxicity. The source of contaminated soil used in the study was from a leak in underground storage tanks and pipelines in one of the Libyan local refineries. Objects of the study assess effects of nutrient addition in TBH transformation, determine the presence of hydrocarbon degrading genes in microbial communities in soil samples, pilot-scale bioremediation of real contaminated soils using methods optimized at lab scales, examine the potential to use plants to reduce residual levels of TBH remaining after bioremediation, determine the effects of different ozone exposure duration times on hydrocarbon transformation in soils. The contaminated soils were transferred to the site of the field trial, and the average initial TBH concentration of the contaminated soil used in this study was in the range of 20,733 to 22,366 milligram per kilogram. An additional portion of the contaminated soil was diluted by using a clean soil to an initial contaminated soil TBH concentration of 16,000 to 17,266 milligram per kilogram. The setup of land farming treatment unit for on-site field studies were used to monitor the bioremediation of contaminated soil. Around 0.5 tons of soil mixture was spread to a thickness of approximately 20 centimeters in 50 land treatment unit plots. The land treatment units were divided to 5 land treatment units. Land treatment unit 1 contains contaminated soil treated with urea ratio 100:1. Land treatment unit 2 contains contaminated soil treated with urea mixed with NPK ratio 100:1. Land treatment unit 3 contains contaminated soil only and works as a control. Land treatment unit 4 control contains contaminated soil diluted with clean soil. Land treatment unit 5 diluted soil treated with urea mixed with NPK ratio 100:10:1. Urea is a popular nitrogen soil for enhanced biodegradation because it's high nitrogen content and slow nitrogen releasing characteristics. So the soils inside the land treatment plots was manually mixed up to a depth of 20 centimeters to maintain a homogeneous mixture, and water holding capacity was adjusted to 16%. So water was added to land treatment units once a week.
It seems that is feasible bioremediation also in country where you don't have much water to use. Natalia, the floor is yours.
Thank you, Marco. Indeed, it seems a very promising approach for remediating the residues of hydrocarbons. Now I give the floor to Jyoti to present the last speaker of today. Thank you.
Yes, so it is my pleasure to introduce the final speaker of the session, Jason Verginelli from the University of Rome Tor Vergata. And his topic is estimating natural attenuation rates at hydrocarbon contaminated sites using closed and open flux chambers. Over to you, Jason.
Okay. Hello, thanks Jyoti for the introduction. So in my presentation, I will focus on some quantification methods that can be used to estimate the natural attenuation rates at sites contaminated by petroleum hydrocarbons. Now, as you know, petroleum hydrocarbons in the subsurface are subjected to a series of phenomena that can lead to a substantial reduction of the mass of the contaminants in the source zone and in general in the subsurface. Now, for petroleum hydrocarbons, the two primary natural attenuation pathway are the so-called groundwater pathway, that regards the dissolution of the contaminants of petroleum hydrocarbons from the source zone, from the nipple body to groundwater, with the subsequent biodegradation as dissolved phase. And this is a pathway that is known since early 90s from the experience gained from monitored natural attenuation. But this is only one of the pathway. Indeed, there are other pathways related to the volatilization that were recently found to play a key role. These pathway regards the methanogenic activity that can occur within the nipple body and the emission of methane and other VOC that then can be degraded in the unsaturated zone. All these processes together are usually indicated as natural source zone depletion. What was found in a recent year, let's say in the last 10 years, is that the volatilization pathway that till 10 years ago was ignored actually can reach up to 90 to 99% of the overall source depletion. This means, in other words, that what we thought till some years ago were the natural attenuation rates resulting from the monitored natural attenuation techniques were actually underestimated of at least a factor of 10 or 100. And indeed, some recent works, for instance, this is a work, a review of GARG of 2017, highlighted that the natural attenuation rates, considering both the volatilization pathway and the groundwater pathway, can reach rates in the order of 1 to 10 kilograms per square meter per year. So we are talking really of very high numbers that are comparable with the ones that can be achieved using active remediation systems. So in this view, now there is a growing interest in developing methods that can be applied to quantify the natural source zone depletion rates at sites contaminated by petroleum hydrocarbons. Here in this slide, I have reported the methods most widely applied for this topic. And in particular, the first method that I have reported is the so-called gradient method that is based on soil gas sampling at different depths that allow to estimate a flux of oxygen or CO2 that can be converted in a natural attenuation rate. Alternatively, CO2 surface measurement can be performed using passive traps or using a flux chamber. That is the focus of my presentation. The flux chamber are actually inverted containers that are placed into the surface and in which the vapors emitted from the subsurface are accumulated over time. Then, based on the VOC and CO2 accumulated inside the chamber, it is possible to use different types of flux chamber to estimate the fluxes. For instance, the types of flux chamber that I have considered here are the so-called dynamic closed chamber, or DCC, that are equipped with a gas analyzer using a PID or an infrared sensor that allow to measure continuously the concentration of total VOC and CO2 inside the chamber. And based on the increase of the concentration inside the chamber, it is then possible, using the Fick's law, to estimate the flux of CO2 or total VOC. This is one method, very, let's say, cheap and very fast. The only limitation, or one limitation of this method, is that this method does not allow to estimate the concentration of each VOC. To have the VOC speciation, you can use the so-called open flux chamber. In this case, in this chamber, an inert gas that usually is nitrogen is fluxed into the device in order to establish a steady state condition. When you reach a steady state condition, the air inside the chamber can be sampled and analyzed for each specific VOC of interest. Then, by knowing the concentration of the VOC inside the chamber and by knowing the flow rate of the inert gas, it is possible to convert this concentration in a flux. So these methods are very widely applied, at least in Italy, for risk assessment, but they can also be used for quantifying the natural source zone depletion. So here I will show you two simple methods that can be used, and I will start with the dynamic closed chambers. In this case, to estimate the natural attenuation rates, some flux chamber can be placed above the source of contamination, and in this way you can measure the total CO2 emitted from the source zone, and then to correct this total CO2 for the CO2 that can be produced as a result of natural soil respiration, you need to place some flux chamber also in an area not contaminated by petroleum hydrocarbons. So in this way, you can correct the total flux of CO2 above the source zone in order to estimate the CO2 that can be ascribed to the only biodegradation of petroleum hydrocarbons. Then, by introducing a stoichiometric coefficient that is representative of a mineralization reaction, you can convert this CO2 flux in a natural source zone depletion rate. So the method is quite simple. Here I show just an example of the application of this method to a site contaminated in Italy by petroleum hydrocarbons. Here I have reported the average CO2 fluxes measured in different areas of this site, and also the average CO2 flux measured in an area not contaminated by petroleum hydrocarbons. You can see that the total CO2 fluxes are higher in the areas with the presence of hydrocarbons, and in particular in the areas with the presence of free nipple. So this is an evidence of natural attenuation. So by applying the method that I showed you before, we converted these fluxes in a natural attenuation rate. And here are the results that we have obtained for the different areas and for the different campaigns. You can see that the numbers that we have obtained are quite high. We're talking of numbers in the order of 1,000 grams per square meter per year. So very high number that are also comparable to the average values that I have mentioned before are reported also in the literature. So this is the method for the DCC, so relatively simple. Another method that can be to estimate the natural source zone depletion rates is the use of this open flux chamber. In this case, the open flux chamber can be placed directly above the source of contamination. So in this way, you can have an information about the emission of each VOC of interest. And then by comparing these measured fluxes of each VOC with the flux of VOC expected using a diffusive model starting from the concentration detected in the source and by ignoring biodegradation, by difference you can estimate which are the natural attenuation rates ascribed to biodegradation. So also this method is quite simple. Here I show you another example of application. This is another site contaminated by petroleum hydrocarbons, always located in Italy. Here I will focus on the flux chamber that we have installed in this site. And here, first of all, I show you the results that we have obtained in terms of comparison of the flux of BTX expected using a diffusive model with respect to the flux measured with the chambers. You can see that most of the data fall above the one-to-one line, and in particular you can observe that in most of cases the non-reactive model overestimated the emitted flux up to over 3 orders of magnitude. So this is an evidence of natural attenuation in the substrate phase. So using the methods that I described before, we have converted this information in a natural attenuation rate. And here are the results that we have obtained for the different sampling points. You can see also in this case that the numbers that we obtain are high, relatively high. We are talking here in many cases of numbers that are in the order of 500 to 600 grams per square meter per year, that again are similar to the values reported in the literature. So in conclusion, based on our experience, and of course based also on the findings of, reported in the recent literature, these natural attenuation rates, considering both the groundwater and volatilization pathway, are really high numbers, rates that are comparable with the ones that can be achieved using active remediation systems, in particular for the late stage of an active remediation system. And so we believe that in the near future, natural source zone depletion should be considered for a proper management and design of remediation at sites contaminated by petroleum hydrocarbons. Thank you.
Thank you very much, Jason. That was a very interesting presentation. I think I will pass it on to Frank now to open the next session.
Thank you very much, Joy, to giving me the word back. Here in Amsterdam, enthusiasm really splashed on the screen. It was very interesting presentations, very stimulating. Not only that you learn a lot about bioremediation techniques, but you also get inspired. I got some ideas, some research ideas myself. So I'm very happy with the session. Thank you very much for all the 6 speakers. Let's open the panel discussion. I have quite some questions myself. I'd like to start with Yanni. And first, I'd like to ask my co-chairs, Josja and Nathalie, do you have some questions for Yanni on the phyto extraction?
Yeah, well, there were some questions on the chat, but I think Jejani already responded to them. And I also have one question for you. I wonder, because you conducted this experiment at the field level, so I wonder if you perceive a good acceptance of these nature-based practices or remediation techniques by governmental and industrial actors.
Sorry, Natalia, can you repeat?
Can you hear me?
Question? I couldn't hear. Yeah.
Let me try with the microphone because my computer. Can you hear me now?
Yeah, now it's clearer.
Is that okay? Okay, sorry, my computer is having some issues. So my question is that since you developed this trial at the field level, I wonder if you perceive a good reaction or acceptation of these nature-based solutions by governmental and industrial actors.
Yes, I think it is a good solution. It is cheap, but the only shortcoming would be it takes time. You know, when you are planting trees, you have to wait. And as you see that what we did was that we introduced the nurseries first. After that, we decided to introduce the endemic species. And this is not the only site that we are working at. We are also looking at an ex-gold mining area and putting in similar strategies. And we will be sharing the results probably in the upcoming conferences later.
Okay. I have a question for Jejani and Chuck combined. because you both had these presentations about phyto-extraction. Of course, phyto-extraction is a very elegant nature-based solution. I only wonder, can you tell me something about the time that it takes to remediate your soil or to get your soils at an acceptable level? Because that's often the problem with phyto-extractions, I believe. Maybe you start, Yanni, and maybe Chuck also could say a few words about that.
So thanks, Frank, for the question. As I presented just now, when we planted the trees, they were about 3 months old. Then we were only able to go and collect the samples like after 5 years, and we saw that the amount of metals, the elements were quite high. So we are doing periodic analysis and data collection. The next one would be coming in November. So we are hoping that we are assuming that the levels would have been reduced by then after a gap of another 5 years. For now, we don't have the exact figure on how many years it takes to actually remove all the contaminants from the soil.
And if I ask you, do you dare to make a rough estimate? Is it 10 years, 20 years?
I would say it will take about 10 to 15 years, because depends on the species as well, Frank, because we are only testing this one species. We have not tried the other species. So probably different species may have tendency to different rates of absorption.
Thank you so much, Yanni, for this clear answer to these questions. Let's move to Chuck. Chuck, you also have an answer to my question?
Yes, Frank, thank you for the question. And I have to echo what Gianni have said, and I agree that it really depends on what type of plant species that you use. And theoretically, I think Gianni have tried the on-site treatment remediation, so that will be more accurate in the sense that most of my works are on the laboratory base. So it can be biased, and also because we are doing artificial spiking, and of course the contamination and accumulations are more happening where it accumulates faster, and that is something unavoidable. But in real situations, the accumulation maybe take slower time. But theoretically, even though we have periodical assessment on the soil, on the ground, or even on the plants itself, we are never able to have a certain final answer to determine that it is completely remediated, because we would not know how bad the condition is,
Of course, I understand that very well. So yes.
So there's always a limitation in phyto-remediation in other way. Okay.
Joyte, Natalia, you have any further questions for Chuck? If not, I have.
Well, I have one for both of them, also dealing with phyto-remediation, because one of the challenges also of the phyto-remediation is what to do with the residues of those plants that accumulate the contaminants, mainly not only in the roots, but also on the shoots and the foliage. So what are you considering this part of the phyto-remediation process? And in case you are, what are you planning to do with those contaminated plant materials?
So there have been some studies reporting on phyto-mining, Natalia, but at the moment of time, we have not tried it fully. What we do is that we bring back whatever that we have collected from the site, and we do send it into the, we have some small-scale industries who reuse it for pulp and paper at the moment.
Well, and for my sites, normally it is hazardous, and we have to categorize it as a schedule waste, which is hazardous. So we will either send it to a proper sanitary landfill, or we to incinerate it. But technically, as what Jenny have said, I also want to second that normally in our field of phyto-remediations, there are studies that if it's essential elements such as gold or even silvers, you know, we might do further extraction. I think Gianni know the field very well since she and I work in the same field of works. Yeah, but in the current market, there are some researchers are working into that. They are possible to extract those essential elements.
I have an additional question for Chuck. I was very much interested that you made a comparison between lead and cadmium extractions. I expected, of course, that cadmium would be more successful than lead because lead is not as much as available as cadmium. I believe that your conclusion was that lead did quite well using nitrogen fertilizer. But in absolute terms, is it possible to remove a significant amount of lead by phyto-extraction?
Well, theoretically, nitrogen fertilizer in my research results propose that it is good, but EDTA works well under both conditions. So in short, based on the research findings, I would not recommend N fertilizers to be single-use soil amendments. But as far as my concerns, you know, I did the studies for several years, including present with both cadmium and lead. Actually, lead tends to accumulate more and compared to cadmium. And of course, I'm currently working not just in cadmium and lead, I'm also starting to work on arsenic and also chromium at this moment. Yeah, about to start a new project. But in short, I would say that N fertilizer would not be an ultimate soil amendments for this case of remediation.
But also to significantly reduce your concentrations in soil for lead, that's possible within 1 or 2 decades?
Well, I don't think it will be an ultimate choice. I would often prefer, even though I'm working on phyto-remediation, we often recommend incorporations of physical, chemical, and also biological treatments, if possible, in order to speed up the process at one go, at the same time to also to overcome other limitations as we do observe in phyto-remediations. That is the ultimate outcome that we want to achieve, I think, end of the day. Sorry, Frank, I think you mute yourself.
Thanks, Chuck. Thanks for helping out. Are there any questions on the chat for Jenny or Chuck? Yes,
there were. There were, but I think they have been already solved.
They've been answered. Huh?
Yeah.
Okay, let's move to Saida, who also gave an excellent presentation on the irrigation channels and the microfibers. Josia or Natalia, you have some questions for Saida?
Yeah, I'm here to answer.
Yeah, I look forward to your questions. Yeah.
I have.
You have one, Natalia, please.
Yeah. The proposal of using a biofilm, it was really surprising, and I think is very promising. But I wonder about the cost effectiveness of this proposal at large scale. Have you made some estimates of how much it will cost and if it will be effective to apply it to the whole system?
I like your question. That's the first thing we did actually, because without that estimation, we could not do anything. So the components of it is not very expensive. We have fermenter to make the biofilm, so it's easier to make the biofilm. Active sludge available a lot. And the microfiber that we're going to use is not expensive one. This is a cheaper one. The only actually the challenging part is that, you know, technological application of that, you know, where we go and, you know, cover all these canals, and that's a little bit more expensive. But when we talk about the circular economy and when we see the results of that, so when we actually have the difference in between the what we get out of that, then we can see that we definitely will win. We're going to go in plus instead of minus. So we can promise to the company industries where they would apply it that they will spend some money, yes, and it's, I don't want to mention the numbers because it would be different in other countries, right? And it will be my own currency, so a little bit hard to change the currency immediately. So we estimated that, so we can get twice more money if we apply this technology, twice more. That's what we can say. And definitely, if we talk about the green circle, green and circular economy, then this is the best way. This is the best way, and we can also help, not only get money, but we can also help the environment. Thank you, great question.
Thank you so much.
What will be your next steps, Saida, in this research, or is it finished now, your project?
We, in the laboratory, we continue the experiment because challenging just outcoming all the time. And we are working, as I mentioned, we're working on the special computer program that would help us to estimate, because now we're just doing like hand estimation, which is not useful. So the computer program that would estimate application of these biofilters in the real conditions. So like, for example, the wind, and also we will use the GIS system, so we can actually evaluate what's going on using the GIS system. That would be differently. So we use ArcGIS 9 for this purpose, but it's not solved yet. So we're just working on that. And the next step will be begging the industry to buy it.
Ah, of course. Yeah, that's very smart. That's very smart. Okay, thank you, Saida.
Thank you too.
Another enthusiastic presentation from Alexandru, a lot of information. Very enthusiastic, very energetic. Do we have questions for Alexandru? I saw some questions on the chat, but maybe we start with Natalia and Josjoiti. We have some questions, or do we go to the chat?
I did have one for Alex. I know that in Copenhagen, it's really drinking water is from groundwater. I know that pesticides is a big problem in Copenhagen. So I was just wondering if there is that sort of significance in your project. Is that why you're looking at degrading pesticides for a change?
Yes, thank you very much for the question. It actually fits very well. Yes, to begin with, this whole project started as a collaboration with FMC Corporation. And most of the soil is not from Copenhagen. It is from the Cheminova plant, which is on the other island, because there is where the pesticides were manufactured. And as we all know, so the best place to get organisms that are supposed to degrade something would be to take them from a place that has a lot of that for a long period of time, so they get adapted to utilizing it. And yes, indeed, DICAMBA is one of the pesticides that we use. The others were in insecticides mostly, but they were also non-water soluble. DICAMBA, as I said, is very water soluble, so it has this very, very big risk of actually drifting. You will not find very high concentrations of DICAMBA in soil because it tends to just run off. And that is a big issue that we're encountering here also because we have multiple water sources in Copenhagen. But that would mean that still we would need to take very close care of how we're handling our pesticides waste, which I cannot say in the world that this right now is done very properly anyway, because we don't even know how to get rid of them. So it could be this microbial degradation, this consortia or pure culture that maybe could counter. And this is why we're also trying, because if we also have the different function, if we can also help the plants and the crops, we can convince people to better utilize it. And it would turn it into more of a product than just a theory. So yes, thank you very much for the question.
I'm checking the chat. I think I saw some questions for Alexander, but I cannot find it back. No, maybe not. I do not have any further questions for you, Alexander. You, Joiti or Natalia, any more questions?
Well, I have like more general question for Alexander, but anyone else can reply if you consider it. One of the problems of the pesticide contaminated soils are the microorganisms and also plants are affected by those pesticides. So do you think in
soils highly affected by those pesticides? In your case, because you say this pesticide tends to leach quite easily, so maybe the populations in the soil are not affected, but what happened with other kinds of pesticides that remain in the soil? Do you think that phyto and bioremediation are feasible techniques for those kind of contamination?
Yes, thank you very much for the question. There are a few things to consider first. I would say that also it depends very much on the pesticide's availability to the organisms, of course, so the solubility, but also on how easily degraded it is by abiotic factors as well. So for example, if a pesticide keeps its structure like intact, whatever, so regardless of temperature factors or water implication, that indeed would mean that it doesn't interact very well with other organisms. But for example, I've read this about because I was working with malathion as well. And malathion can turn into a compound called isomalathion just by being exposed to some temperature changes. And that is so much more toxic than the original. So already you're changing the populations just because only the very resilient organisms would be able to survive such a recalcitrant compound. Taking that into consideration as well, DICAMBA is easily taken in by organisms. It is easily degraded because some of the bonds are, of course, easier to break, especially the ones that are the chlorine bonds, easier to take in. So that would mean that the organisms have an easier time of consuming it. And of course, while you put them in the soil, the pesticide application starts selecting for some organisms. So it does affect the soil microbiota greatly. And yes, I think bioremediation of many types, including biostimulation, so just stimulating the present organisms, could be a very nice solution. I have taken the different route, and what we're doing is we're applying organisms from a different site to that site in order to test, because we know specifically what their functions they're gonna have.
Okay, unfortunately, we are not able to ask questions to Altab Hakimia. So we go to the final speaker of the session, Jason. I'd like to start the first question for you, Jason, if you don't mind. You showed the possibilities for dynamic and open flux chambers. When I want to use one of these two procedures in the field, how can I decide which one to use? Do I need to call you, or are there some criteria on which I can decide what is the best option for my case?
Thanks for the question. Yes, actually, surely dynamic closed chamber are more economic and more fast. So for to quantify the natural attenuation rates, perhaps closed chamber are better. But if you want to have an information about the concentration of DOC that can be used also for a risk assessment procedure, you have also to apply an open flux chamber. So it depends, of course, on the information that you want to obtain from the sampling.
And also from the money you can and like to spend, of course. Eh?
Yes.
Yeah. Okay, thank you. Maybe Natalia or Yoiti, some questions for Jason?
I have a quick question, or Joyty, you can
No, go ahead, Natalie.
Okay, thank you. I just want to know how easy is to determine where to put the control chamber, because if you have an apple, you have to do some tests to see which area is covered by this nipple and then decide where to put the control one. So is this quite complicated? It was costly. So I was just wondering about that.
No, I see your question. I see your point, and thanks for the question. Yes, of course, you need to first have a characterization, a good characterization of the site. So you need to have groundwater wells. Surely now I show just a method to quantify the natural soil respiration rate. So to understand how to where to place the blank sample. But there are other techniques. For instance, there are other techniques based on radiocarbon analysis that allow just with one chamber to understand which is the contribution of natural soil respiration compared to the biodegradation of petroleum hydrocarbons. So there are different solutions, of course, with different costs. What I presented is the more fast and economic, but of course, there are other alternatives.
Jorti, you had a question?
You are mute, Jorti.
I'm mindful of the time, Frank, but maybe just a quick one. Did the methods that you show take account of natural attenuation in the groundwater?
Thanks for the question. Yes, actually, you're right. The methods I presented are actually only focused on the volatilization pathway. So in this view, the rates that I showed are a bit underestimated. But at the same time, I showed that the volatilization pathway is actually the 90 to 99% of the overall attenuation. So in this view, are only slightly underestimated the natural attenuation rates obtained with this method. So, thank you.
Okay, guys, it's 11 o'clock. We're getting towards the end of the session. However, I have one general question for all 5 of you. As an ending of this session. Imagine I have a sustainable amount of money for you. I have a sustainable budget. It's hypothetical because I don't have it. And I met you here at Remtech as a team, and I think that looks good. I ask you as a team to start a project on bioremediation. What would be your desires? What would you do? What are the most important issues you would like to pick up if you have a lot of money to work on bioremediation? Any of you who like to answer, please give an answer to this question. Don't tell me you don't have any desires on research on bioremediation.
Then, if I may.
Sure, Alessandro, go ahead.
To be fair, I would go with the original idea in my field at least and try to expand this into a product. So something that I would be able to actually apply actively on crops. To create a dry product. That would have been the end goal, to create a dry product to be able to apply on the crops, and that would provide better food at the same time as plant protection. And that would have been the end goal if we were to be speaking from a corporate point of view, so to speak. If I would have funding for this, that would have been the step to continue this until it would turn it into a product that would be easily applicable and transferable in any part of the world.
Sounds interesting, sounds interesting. Yeah, Yanni, Chuck, any desires for research? Or you're all happy with your own research?
Well, for me, Frank, as I said before, it is a long-term study if we want to use trees in order for phyto-remediation. My personal, intention would be that could actually train up somebody after me to continue monitoring whatever work that I've been doing, rather than it has been stopped halfway and there is enough funding for us to continue this project for long term, rather than the project has to be ended due to the funding has been ended.
Okay.
Well, I think I would just quickly say that I would like to expand the research scopes and horizons as well in order to combine multiple remediation strategies in order to speed up the process. I think ultimately we want to reduce or minimize contaminated areas. So that's what I think I will do with the fund if it's granted. Thank you.
If it is granted, okay. Saida, you're the final word is to you. Saida, you have some desires, some plans to spend this money, or are you happy with your research, what you're doing right now? Is that enough?
So what I think that since you, Frank, the one who has this money, I would first learn what is your interest area to clean up. And then I will suggest you all of those bioremediation methods for choose. And then whatever you will choose, we will build the joint project to do that. And actually, I like the phyto-remediation methods that my colleagues presented. What I will definitely go with, you know, I will merge my methods, I mean, not my colleagues and my method together with this phyto-remediation method. I would say this would be more effective than only the method that we developed. And definitely we would, you know, we will try to make you happy because you're going to spend the money for that. So we need to do it jointly, definitely.
That sounds good. That sounds good, Saida. Okay, I think we have to end this session. It's almost 7-8 minutes past 11. I think we can be very happy about the quality of our session. I'm very happy with the session. I learned a lot. I hope you all agree with me on this. Thank you very much for being here. And the last thing I say is that I like to point out that the next session is at 11:30 in half an hour, and that will be on innovative characterization technologies and digital innovation. I hope to see you back then, and enjoy the rest of Rentec Europe 2020. Bye-bye. See you again.
Thank you. Great job. Thank you very much.
Bye, bye, everyone.
Thank you to everyone.
Bye.
Thank
Bye,
you,
Frank.
Jyoti and Natalia and all speakers. Thank you very much. Interesting.
Thank you.
Thank you, Mark.
Thank you.
Thank you.
Thanks.